Okay, let's review DNA and RNA. If you remember, DNA is short for deoxyribonucleic acid. So I highlighted and underlined where the D, the N, and the A come from for deoxyribonucleic acid. If you look at this diagram here, you can see this is a model of DNA. And hopefully you're able to identify the three parts. We have our sugar, deoxyribose, and remember, sugars typically end in OSE, so that gives you a clue that that is indeed the sugar. Then we have our phosphate backbone. As you can see, the phosphates actually hold and link all of the sugars together. And you can see there go the phosphates linking the sugars to one another, like that. And last but not least, we have our nitrogenous bases. We have A, T, C, and G. Remember, A stands for adenine, C stands for cytosine, G for guanine, T for thymine. So those are the four bases that are found in DNA. Now, one sugar, one phosphate, and one base. What do those make up? Remember, if we have a sugar, phosphate, and one nitrogenous base, such as that, remember that makes up a nucleotide. Now let's move on to RNA. RNA is ribonucleic acid, so it's a little bit shorter than what DNA is. The R, the N, and the A for ribonucleic acid. In the picture, you can see that unlike DNA, RNA is single-stranded, so we only have one strand. The sugar found in RNA is ribose. Remember, OSE. And instead of thymine, RNA does not have thymine. RNA has uracil. So RNA does have all of the other bases. So RNA has A for adenine, cytosine, G for guanine, and it has U for uracil. So this time, because we already know that C and G, those are corresponding base pairs. Now, since this is RNA and not DNA, in this case, A doesn't pair with T, A will pair with U, and U will pair with A whenever we're dealing with RNA. So your clue, you'll always know that you're looking at a strand of RNA if you see the U, because it's uracil. And we make RNA from the process transcription. So let's get ready to make some DNA, and then we'll go ahead and make some RNA. So we'll start with DNA replication. And remember, DNA replication, that's how more DNA is made. So this strand that we have that's already printed here, this is going to represent our original strand. Okay, so now we have to come up with our complementary strand. So we have A, which pairs with T, T pairs with A, G with C, C with G, C with G, T with A, G with C, T with A, C with G, T with A, G with C, and A with T. So the blue will represent our complementary strand. From our complementary strand, we can go ahead. So now that we've replicated DNA, so we now have our new strand of DNA, now we can go ahead and make our RNA. And we'll start from using our complementary strand. So T, we have A, now we have U because we're making RNA. C is G, G is C, G is C, A is U, C is G, A is U, G would be C, A would be a U, C would be a G, and T would be an A. So we've just made our strand of mRNA. And we're going to need to utilize this strand of mRNA to figure out and determine the protein. So in determining our proteins, we have to use codons. And remember, codons represent a group of 3 nucleotides. And although we don't see the phosphate and the sugar, we do know that they are attached to that. So let's go on and prepare to utilize our codons. In this case, this is our first codon, second codon, third, and fourth. So all of these will represent the codons. And we're going to use the codons to determine the amino acid production. So we do need to use our handy little wheel, and I will re-record our codons. We had A, U, G, C, C, U, G, U, C, and U, G, A. So let's use our wheel to find the corresponding amino acids. I have AUG, so it's A, U, and G, and that codes for methionine. And that's typically our start codon anyway. And for short, we'll just say MT. Now let's look up CCU. Our first one C, second one C, and U, which gives us proline. So we'll say PRO. Next codon is GUC. So let's see, we have GUC, which gives us valine. And finally, our last codon, UGA, UGA gives us stop. That is our stop codon. And remember the stop codon, that just lets us know that that is the end of protein synthesis for this particular strand. And we've gone through DNA replication and protein synthesis.